I'd like to get started. I'd like to welcome people to the March 16th meeting of the MSM IMAG Multiscale Modeling and Biopandemics Working Group. We're very glad to have our speaker today talking to us about seroreversion for assessing SARS-CoV-2 seroprevalence, which is a follow-on to some talks we heard a long time ago, which I think were quite intriguing for people. As always, Tom Helikar and I are available for comments, questions, and suggestions. Jim Sluka, Lorenzo, and Joel are available to help as well. Specifically, if you have any web issues, go to Jim. If you have any suggestions for activities and coordination, please reach out to Lorenzo. We have our communication channels. They only work if people use them. Please help us use them. In particular, the IMAG MSM wiki page is always in need of new material. We have a LinkedIn page that could use posts, and we have our YouTube archive, which now, as of today, will have 103 videos on it. We have the handle MSM Viral. If you tag that, you may be able to help us reach out to a bigger audience, and we'd appreciate your support. I'd like to remind people of the upcoming Immune Digital Twins workshop in Saclay from the 15th of March to the 2nd of June. Many of us are already planning on being there. Please remind your grad students and postdocs that this is a meeting that is open to people at all stages of career, and we'd appreciate your suggestions of people to attend. There's going to be the annual IMAG meeting in June in Bethesda. You can contact Herbert Sauro and Shane Pierce-Kotler for more information about that, or Grace Peng also. I'm always advertising the fact that at IU we have 2 open positions in biomedical digital twins. That search is underway. We would love to have people that you would recommend apply for that. These can be open rank positions, anything from starting assistant professors up to chaired professors. Something that just came across my desk today, which I wish I had known about earlier, is that the Academy Health has issued a call for pre-proposals. Unfortunately, they are due March 21st, but they're short proposals, asking for grants related to computation to reduce diagnostic delays in acute infection, which is certainly an area that we would like to see immune digital twins used in. They have a very nice white paper laying out the issues of the costs of delayed diagnosis. They have 3 focus areas, one of which is cancer, which potentially could have an immune component, one of which is acute cardiovascular events, which is probably a little bit less in the direction of the people in this working group, and the third one of which is sepsis diagnosis, which is very much the kinds of things that people here are interested in. Even if none of us are going to apply for it, I think the white paper lays out a compelling case for the role of digital twins as a mode of diagnostic and prognostic to improve health equity. And so I think even if you're not going to apply for that, you might find the white paper that they've written quite interesting. If anybody does want to apply for it, I'd be happy to talk to you about it. Tom Helikar has been working very hard on developing the Glimprint website. He's updated the website, and we would be very grateful to you for your input. I hope in the steering committee meeting tomorrow we can work on that a bit. We are trying to make it into something that we can all be proud of. To begin with, of course, it will have very similar content to the IMAG MSM webpage. As time develops, I hope that these websites complement each other and we'd be looking forward to working on developing that. Tom, did you have any comments you want to make about that?
I just want to point out right now, if you go to the website, it's still the underdeveloped version, but all of the content right now is on the staging version of this site, and it should go live sometime this afternoon. So
people's feedback and suggestions would be most appreciated for that. Okay. We will have a steering committee meeting tomorrow at 11 a.m. If you think you should be invited and you haven't been, please let us know. And now there's an opportunity. Any other announcements or comments before we get started on the seminar? Okay, coming up, we have unexpectedly an open slot on March 30th. So Lorenzo will be reaching out to people to try to fill that slot. April 13th, we have Mikhail Benson talking about the Swedish Digital Twins Initiative, where it's a very big project there with a lot of government support. And then April 27th, we have Claudio Montero again from Nova Discovery. Will be continuing his exploration of the wonderful software platform that they've been developing for developing in-host and cross-population virtual patient models for in silico clinical trials. If you have suggestions for speakers, please let me, Lorenzo, and Tom know, and we'll do our best to get them on the schedule. Okay, today we're very pleased to have our speaker from Case Western, talking about serology and seroreversion, modeling seroreversion for assessing SARS-CoV-2 seroprevalence. And I will turn over the screen share to you.
So thank you for having me. My name is Owusu-Boaitey. My background is in immunology, and a lot of the collaborators on this project bring other expertise from modeling to epidemiological analysis to or just other aspects of understanding pandemics. So I just wanted to give credit to them initially. The focus of this presentation will be seroreversion or waning of antibodies after infection, but I'll also touch on some of the other data we presented on the topic of serology. And the basis of this paper is a recently accepted manuscript from our team at Seroprevalence. And these are some of the other collaborators we've been working with that are not co-authors on that paper. Again, my background's in immunology, so it's very nice to have a group of collaborators who bring experience from other fields. Again, especially statistical modeling and just understanding epidemiological data. So just for context, for decades, we've used serology to assess prevalence of infection, dating at least back to the HIV pandemic. This is particularly useful for the COVID-19 pandemic because data suggests the vast majority of individuals seroconvert after infection, enabling us to use antibody prevalence to approximate prevalence of past infection. Once we have these serology-based estimates of prevalence, we can use that to estimate other metrics such as fatality risk following infection or IFR, and then hospitalization risk following infection or IHR. Our work is mostly focused on IFR, but we have upcoming projects assessing IHR as well, and some of our collaborators have also published on IHR. This is an improvement over case fatality risk, which is limited by how well PCR testing is being performed or rapid antigen testing. With serology, you're able to capture infections that may have occurred weeks or months ago, but with case fatality risks, you're only able to capture fairly recent infections, leading to underestimation of prevalence. So the CFR is more useful if you're concerned with things like incidence as opposed to prevalence. So modeling prevalence using serology has many moving parts because, especially in the context of estimating IFR, because you need to both capture the number of infections and accurately capture the number of deaths while having appropriate lag times between them. So when modeling infections, we have to deal with things such as sensitivity and specificity of tests, so adjusting for that, and making sure to sample a appropriate sample frame using random sampling. Convenient samples of blood donors and other populations can lead to inaccurate prevalence estimates. Since there's also a lag period between infection, then seroconversion, death, and reporting of deaths, we need to model those lags appropriately. And also keep in mind over and under reporting of deaths, because those can skew fatality rate estimates as well. There are other less recognized aspects of modeling that may be a concern, such as migration of infected individuals, especially early in the pandemic. So, for example, if prevalence is quite low, like say 1%, migration of say half the infected population to another area can sharply skew prevalence. And there's also risk of extrapolating studies of local populations to national populations if the local population is necessarily representative of what's going on nationally. So in our prior work in 2020, we used seroprevalence studies, both convenient samples and random samples, along with areas with heavy testing to estimate IFR. We adjusted for uncertainty and specificity using a Bayesian model. We had about a 4-week lag between serology, midpoint of serology testing and reporting. We use a combination of national and local studies. And we focused on high income locations because they were likely to have sufficient mortality reporting as opposed to say areas in South Asia or Africa where COVID-19 deaths may be underreported. So that was our 2020 paper, and those estimates form the basis of the CDC's IFR estimates that they use for pandemic planning. So looking at this figure from our paper, you can see there is some variance in IFR, but we argued in that paper that you can fairly well predict the IFR for a given region just based on the prevalence and the distribution of age. So basically the distribution of infections across ages. So the basic argument was that IFR in these regions is fairly consistent age-specific IFR, and what differs is just the age distribution of infections. And that IFR was fairly similar in locations that had sufficient testing like PCR testing or rapid antigen testing when this was compared to IFR from seroprevalence. But there were some outliers in this analysis, as you can see for Italy in the corner there. Italy's IFR is a bit higher than expected. And this has been noted by other researchers such as Don Ioannidis. And the possible explanation for this is seroreversion or antibody waning with time. So I think everyone's familiar with the basic immunology, just as a quick refresher. Following infection, antibody titers increase within 2 weeks of infection. And then after that peak antibody titer, there's a decrease in antibody levels. And depending on the assay, that decrease can go below the limit of detection of an assay. And this is what in our analysis, we call seroreversion, where test sensitivity for a given assay decreases because the antibody titers have decreased below the level of detection for the assay. And this would serve to bias prevalence estimates downwards and therefore bias IFR and IHR estimates upwards. This might matter less early in the pandemic where people have been recently infected, but as the pandemic progresses and there's more time for seroreversion, the bias becomes worse and worse. So it becomes important to adjust for this bias if one wants to perform any longitudinal assessment of prevalence or IFR or IHR. So there are at least 2 methods for assessing seroreversion. One is to perform serology on people with a prior diagnosis, which is diagnosis maybe by clinical suspicion or clinical diagnosis, PCR, rapid antigen tests. The benefit of this is PCR and rapid antigen tests will test someone will test positive relatively close to their infection date. So it allows you to reasonably approximate when they were infected. If they don't remember when, or they don't state when they got their positive tests, one can use modeling to infer the likely infection date from reported case curves. But one downside of this method is it doesn't really account for, one upside of this method is it accounts for cases where someone has failed to seroconvert. So that's assessing seroreversion following diagnosis. Another method is to assess for seroreversion following a prior positive serological test. The downside of this is, since serology can appear positive long after someone's been infected, it's sort of harder to narrow down the infection date. So it basically requires modeling to infer the infection date. And it also doesn't account for cases in which someone has failed to seroconvert, since it's, again, relying on a prior positive serological test. And both of these methods of assessing seroreversion have been employed in the literature. So in our prior work, we focused on our 2022 paper, we focused on assessing seroreversion using prior diagnosis. So not using a prior positive serological test, but instead diagnosis with PCR, rapid antigen test, or clinical diagnosis. And this was focused on lower income nations. The issue with that is we weren't able to do this very systematically. So we lacked data for several locations. So we were only able to perform seroreversion adjustments for seroprevalence studies in some locations. But as you can see, the seroreversion adjustments have a pretty substantial impact on sensitivity, again, ranging from decreases of 70%, 80%, and so on, which can, again, have a substantial effect on inferred IFR and IHR. So in this 2022 paper, which was focused on lower income nations, we made some slight improvements to the previous model we used. As I mentioned before, we were able to do some seroreversion for some locations. We were also able to account for lags, not only by reporting date, but also in cases where date of death was available. So instead of having to roll out just a 4-week lag, we also could have a 2-week lag when there was no lag present from reporting. We also were able to assess locations for risk of underreporting using death certification. So basically looking at the prior decade to see what proportion of deaths in that location were well certified, and also using excess mortality to approximate underreporting of COVID-19 deaths. And in future work, we're going to present some evidence making that adjustment more precise. And we shifted from including convenience samples to just focusing on random samples. So we're making incremental improvements in our modeling, but we're hoping in our upcoming set of projects, our modeling becomes much more refined, as I'll present a little bit later. So the take-home message from this analysis was that once we corrected for underreporting of deaths, population-wide IFR was similar in high-income locations and lower-income locations. That was for population-wide IFR. For age-specific IFR, age-specific IFR was higher in lower-income nations. So the explanation for this is sort of two competing forces. You have age-specific IFR higher in lower-income nations, which would push IFR higher in lower-income nations, but lower-income nations also tend to be younger, and IFR increases with age. So you have sort of those two competing factors that end up balancing out. And also, lower income nations tend to have, less shielding of the elderly from infection. So they have a greater proportion of infection in elderly individuals compared to higher income nations, which again pushes the IFR a little bit higher. So these factors taken together, the age distribution of infections, the median age of the population, and the age-specific IFR, all those factors taken together result in IFR, population-wide IFR being fairly similar in high-income and low-income nations. Sorry. So as I mentioned before, in this 2022 paper, we were only able to perform seroreversion adjustments for some assays, which left room for a more systematic analysis that would enable us to have seroreversion adjustments across studies. So just to comment on prior work, as you can see in the figure on the right, for on the 2020 paper, there is assay-specific seroreversion. So seroreversion isn't just the same across assays. And some prior research also shows that greater disease severity risk, greater disease severity reduces seroreversion risk. But that doesn't really address the question of whether there are specific attributes of an immunoassay that explain why the assay is at greater or lesser risk of seroreversion. This has been a question that's been difficult to assess systematically prior to this pandemic because there hasn't been as much global serology work as occurred during this pandemic. So the COVID-19 pandemic sort of offered a almost unprecedented opportunity to assess seroreversion and how assay characteristics affect seroreversion. So just to comment more on immunoassay design and antigen target. So immunoassays have various designs. There are lateral flow designs that are very useful for very in the field, rapid testing, just quick results. These can be useful in some contexts. It can be useful for people, for like individuals who don't have a lot of expertise and just want to test themselves, but the results tend to be less reliable. There are also quantitative ELISAs that are run in labs, which have more reliable results, but take more time to have a readout. So it might not be as useful to an individual who wants information now. And then there are also neutralization assays that are detecting whether there are neutralizing antibodies present that can help prevent infection. And so these are probably the most clinically relevant immunoassays. And then the immunoassays can target antibodies against various antigens in SARS-CoV-2. So the major focus is on antibodies recognizing nucleocapsid protein, spike protein, and then the receptor binding domain within the spike protein, which is the main means by which SARS-CoV-2 infects. So we wanted to understand how antigen target and immunoassay design impact seroreversion risk. So we performed a systematic review of serology studies that tested individuals following a prior diagnosis by PCR, clinical suspicion, or rapid antigen tests. And we excluded studies that just focused on very particular populations like hospitalized patients or nursing home residents or those vaccinated against SARS-CoV-2, because we wanted to focus on antibodies from infection. And we also wanted to avoid what I previously discussed about disease severity impacting seroreversion risk, because again, hospitalized patients would have less risk of seroreversion. So we didn't want that bias in our results. Our sensitivity was just taken to be the proportion of those with prior diagnosis who tested seropositive. We had a Bayesian hierarchical model to fit to sensitivity with time since diagnosis. And as I continue this presentation, I'll just call that sensitivity versus time of seroreversion curve. It approximates assay-specific seroreversion. And we were able to have results for about 50 assays from 76 studies. And this was, again, recently accepted in Seroprevalence. So some of our results were that some assays, interestingly, showed increased sensitivity with time. So the opposite of seroreversion, which likely has to do with antibody maturation. But sort of another major take-home message was that LFAs, so lateral flow assays, and assays against assays targeting the nucleocapsid antigen were at greater risk of seroreversion. And what makes this particularly useful is that we have seroreversion curves by month, up to 14 months post-diagnosis. So basically, if we have an assay among these 50 assays listed, we can basically tell what test sensitivity is going to be at any month up to 14 months subsequent to diagnosis, enabling us to, in effect, correct for seroreversion. But moving beyond that, there was a question of indirect versus direct ELISAs. Just to give a quick explanation on assay design, so you have your SARS-CoV-2 antigen and for an ELISA just on the plate. You had patient sample serum, and then antibodies present that recognize SARS-CoV-2 bind. And so indirect and direct ELISAs have that same setup, but the difference is how you detect those antibodies. So for a direct ELISA, you're going to detect with SARS-CoV-2 again, which is going to bind to any bound SARS-CoV-2 antibody for detection, while with an indirect ELISA, you're going to be using a secondary antibody that binds to the constant region. So what we found is that indirect ELISAs were at greater seroreversion risk than direct ELISAs. And that's likely due to factors such as better detection of other antibody isotypes, or leading less dilution not needing as much dilution to prevent the secondary antibody from binding to non-SARS-CoV-2 antibodies. So at this point, we knew that indirect ELISAs were at greater risk of seroreversion. Antigen targeting of targeting of nucleocapsid antigen would increase seroreversion risk, and lateral flow assays increase seroreversion risk. So having that information and quantifying the contribution allows us to make predictions of seroreversion curves, the assay where we know antigen targets, whether it's a lateral flow design and whether it's direct or indirect. So just beyond the 50 studies, even beyond the 50 assays we included in our analysis, we can now make predictions of seroreversion for virtually any assay that's used in a seroprevalence study, which enables us to answer questions we couldn't answer before. Because as I mentioned before, longitudinal testing, longitudinal assessment of IFR and prevalence requires correcting for seroreversion. And if you cannot correct for seroreversion with the assay you're using, you're not able to answer certain questions reliably. So just for immediate relevance to prior seroprevalence work, global infection-induced seroprevalence has been previously assessed using serology studies that align with the human immunity protocol. So we looked at the data set for that protocol from SeroTracker to assess how many studies were at higher risk of seroreversion, meaning higher risk meaning lateral flow assays or indirect ELISAs that were focused on the nucleocapsid antigen. And what we found is a substantial proportion, up to a quarter of the data used was at greater risk of seroreversion, but this proportion decreased with time. But it was still fairly substantial even later into the pandemic. So, and this is not something researchers are unfamiliar with. Even in the 2022 paper from the WHO's collaborators, they noted that there was still a need for adjustment for antibody seroreversion. And I think our projects and our results enable that adjustment that they were asking for. Some limitations of our analysis are it focuses on seroreversion following an initial infection. This matters a little bit less in the pre-vaccination period early in the pandemic because prevalence is lower, so reinfection risk is lower. It's unclear how seroreversion varies with age. It's been well established at this point that COVID-19 disease severity risk decreases with age. So that would lead one to think that seroreversion risk would increase with age. Would disease severity risk decreases with age as you get younger. So decrease as people become younger, but that would similarly lead to the implication that maybe seroreversion risk increases as people get younger. We weren't able to assess that because there's very limited data. So that is a remaining limitation of our results. There's likely underrepresentation of asymptomatic individuals in our data set because we're relying on prior SARS-CoV-2 diagnosis, and asymptomatic individuals are less likely to be diagnosed. And we were using case curves to estimate infection trends, not necessarily total infections, but just trends in infections. But this is unreliable, less reliable when very early in the pandemic when testing rates were much lower. So to return to the example I gave before from our 2020 paper about Italy. So we know that the serology study from Italy used an serology study targeting antibodies against the nucleocapsid antigen with an indirect ELISA. So based on our results, that study is at high risk of seroreversion. The baseline sensitivity for that study was reported at 100%. But the seroreversion adjusted sensitivity is substantially lower at 67%, which substantially reduces the IFR estimate for that study. So this is just an example of how our seroreversion adjustments can provide more informative estimates of IFR and prevalence and likely IHR as well, and allow for longitudinal assessments of severity risk. So we are applying this seroreversion framework, the seroreversion modeling framework in ongoing projects. For all the projects I'm about to discuss now, we've only included serology studies that randomly sample a sample frame representative of the general population. So unlike our 2020 paper, we're no longer using locations with intensive rapid antigen or PCR testing, and we're no longer using convenience samples because at this point there are enough serology studies where it's no longer needed. It's no longer necessary to have convenience samples. We focus on the pre-vaccination time period for several reasons. Our seroreversion estimates are again following initial infection, not reinfection, and not following vaccination. So introduction to vaccination looking at a post-vaccination time period would increase risk of reinfection and antibodies from vaccination. And a serology that focuses on antibodies against the nucleocapsid antigen could theoretically be used in a post-vaccination timeframe, but there's evidence suggesting that vaccination reduces the sensitivity of these anti-nucleocapsid assays, making it less viable to apply our model to post-vaccination time periods unless one introduces further adjustments into the model. And also just focusing on the pre-vaccination time period helps us answer certain questions such as the disease burden averted by vaccination, and also how impactful pre-vaccination mitigation strategies were in sort of changing the longitudinal curve of age-specific prevalence or IFR or IHR. So for one of our ongoing projects, we have a model framework where we've implemented our assay-specific seroreversion adjustments with lags for COVID-19 deaths and hospitalizations, and adjustments for test sensitivity and specificity. We're calculating IFR, median IFR and IHR across various time periods and comparing those to early IFR and IHR estimates that were used in pandemic planning. This is some preliminary data using the REACT-2 study of England, which had reported seroprevalence that was decreasing. So there are 4 phases of testing in this study, and you'd expect prevalence, seroprevalence to be increasing with time, but instead it decreases across the first 3 phases, which is either a sign of change in sampling design or likely seroreversion. So we applied our seroreversion modeling framework to this and found that seroreversion sensitivity was decreasing with time, as we'd expect if seroreversion is occurring. If you look at the figures to the right, you can see that, especially for IHR, in the absence of seroreversion adjustments, the later phases of testing have a seroreversion bias that's forcing IHR upwards. But seroreversion adjustment helps correct for that bias. And this is just preliminary data. We're extending this to other locations as well. Some other preliminary data we're sort of aggregating across locations. We see that locations with later dates of serology sampling, seroreversion bias may be likely higher. So the seroreversion bias is causing inflation of IHR estimates. And so this suggests that, as expected, as serology gets later and later in the pandemic, seroreversion adjustments become more and more important for assessing prevalence and severity metrics. So we wanted to more directly assess whether severity risk is changing with time, which to do correctly it's best to have a specific location where serology is repeated multiple times across the pandemic. There's been some prior work on this, trying to assess this, but not with the sort of robust seroreversion adjustments we have across many assays. So we want to assess how IFR and IHR change over time, and whether infection rates, whether prevalence is relatively lower in adults, older adults over time, and whether there's a difference between high income and low income locations in this respect. We also wanted to, in a separate project, we're assessing relative prevalence in children, because there were some suggestions during the pandemic that children were at lower risk of infection. There was some initial data addressing this question, but again, without seroreversion adjustments and with a very limited pool of serology studies. So with the larger pool of serology studies we now have, we feel as if we can better address this question, or more comprehensively address this question. And another ongoing project in which we have collaborators at Northwestern and Harvard that we're working on this with is assessing pandemic burden, specifically infection rates in African nations, because mortality reporting and testing with PCR, rapid antigen testing, and so on, is particularly low in many of these nations. And so having seroreversion adjusted prevalence estimates can help us know how many infections occurred there. And also, if we have locations where we can empirically correct for mortality underreporting, we can provide more robust IFR estimates for those locations, and then use those IFR estimates in combination with other mortality data to back calculate infection rates in other areas. So just to summarize just a lot of this work, serology is very informative during pandemics, as illustrated in the HIV pandemic and also the COVID-19 pandemic. But in order to sort of fully capture that use, seroreversion has to be accounted for as the pandemic progresses. Our seroreversion adjustments based on test characteristics like lateral flow versus indirect versus direct assays can hopefully inform serology testing and seroreversion adjustments during future pathogen outbreaks. Maybe not as much the antigen target, but just the general test design can provide an initial framework for adjusting for seroreversion in future outbreaks before there's much other data to work from. And our seroreversion adjustments will hopefully be important in other ways, such as, again, assessing disease burden averted by vaccination, understanding pandemic burden in areas with less reliable mortality reporting, and assessing age-specific infection risk, and so on. So thank you for your attention, and Looking forward to any questions.
Okay, time for some questions. That went by very fast for me because this is material that's unfamiliar to me. I think it's quite interesting to see your attempt to try to back out real IFR numbers. And I guess the bottom line in that table at the end was 0.3% for over 65s about. Was that
In which table?
I guess at the very end, I was just looking, you had the US data, and at the very end, you had US data, and I see estimated 1 million deaths, 700,000 deaths out of 18 million cases, which would be about a 0.3% death rate per infection. If I understand that table right. For the 65-year plus line there.
Yeah, I think it, for the all ages, it's about 0.6, and I think it's higher for the 65 plus. I'm very bad at doing math.
Did I lose a zero? Did I lose a zero? Was it 3% instead of 0.3%?
Yeah.
1% would be 180. No, so 3%. Yes.
Yeah.
That's 3%.
So for me, sort of the take-home message was that from this table was that IFR and IHR changes of, say, a factor of 2 can result in millions, a difference of millions of hospitalizations and millions of deaths when applied to many infections. So for example, for IFR here, if we just cut IFR by halves with the same prevalence, we'd end up with about 400,000 less deaths. And so when you're trying to do early pandemic planning early in a pandemic, just very slight differences in IFR and IHR matter a lot. And as a pandemic progresses and seroreversion bias becomes more and more important, if you don't correct for that bias, then you're going to overestimate hospitalization risk, you're going to overestimate fatality risk. And once the infections accumulate more and more, that's going to translate into just poor predictions on hospitalizations and total hospitalizations and total deaths. So that's sort of the take-home message I took from this table. Their age-specific IFRs implied from this table match up fairly well with our estimates, even before seroreversion adjustment. So I'm curious to see how well they line up after we complete our seroreversion adjustments.
So I guess you don't have access to serum, so you can't do the tests in a country like China where they're not actually reporting COVID-related deaths in a consistent way. So you'd have to have access. You'd have to have basically access to a sample patient population that was fairly unbiased. I know early on, even Indiana did a bunch of prospective sampling early in the epidemic. I don't know, was that something that was done generally? And I would say picked some set of thousands of random individuals and tested them periodically. I don't know whether there was blood sampling as well, or whether it was just something like a lateral flow or PCR test. But
so that's a fair question. So in this figure here, we do have results from Indiana. It's at the intersection. It's at 0.5 for predicted IFR if you look up from there. And they did report initial testing in April, but it used the same assay as the Italy study, the Abbott Architect assay, which is at increased risk of seroreversion. And I believe they had a second phase of testing they reported in June. So we do have results from that study, and we have performed a systematic review of other studies that have longitudinal testing. There are some locations in Georgia, several studies in England, Spain, Netherlands, and so on. And so one of our projects is, again, assessing how IFR changed in those locations with seroreversion adjustments. So to answer your question, yes, there are places that sampled many individuals over time where we can apply seroreversion adjustments.
No, it's not that often that you see Indiana as a leader in community medicine and the diagnostics. So that's nice to see that. Well, all that I did when you showed it the first time, I didn't see the Indiana doc, so I'm pleased. Could you have any idea what's going on? You pointed out Italy is an outlier on the upside. What do you have any idea what's going on with Portugal and the Netherlands that seem to be outliers in the opposite direction?
That's a very fair question. So for Portugal, we were incorrect in that analysis. We assigned it as a representative study, but upon later reading, it was clearly a convenience sample. We miscategorized it. I believe that is, besides Portugal and New York, every other study was correctly categorized. New York was also a convenience sample. We didn't catch that until after publication. For the Netherlands, there's already data indicating underreporting of mortality, both based on death certificates and based on excess mortality. So that likely explains the lower IFR in the Netherlands.
So let me understand. Maybe I misunderstood this table. So what you're saying here is that if I'm lying under that line and the data are true, that means that the actual IFR is actually reported, is actually recorded deaths. And you're saying that this is it. So if I'm lying under the line, you're saying that I'm fundamentally, somehow I'm underreporting deaths, actual deaths. If I'm lying over the line, I'm overreporting actual deaths.
So that's one possibility. So one possibility is underreporting or overreporting of deaths. Another possibility is that there's an issue with study design. So in Portugal, we think the issue is with study design. For the Netherlands, we think the issue is with, there's independent evidence suggesting the issue is with death reporting. So yes, the line basically, if there's a sharp deviation from the line, it's telling you that there's something strange going on. It can be, again, death reporting, can be study design, it's something. For Indiana, for example, sorry, go ahead.
No, go ahead.
So for Indiana, it's almost certainly due to seroreversion.
Lucas had a question. Can you comment a bit on how cohort tracking could help improve infectious disease surveillance estimates of IFRs and IHRs? And are there any major cohort tracking projects underway in North America and Europe for future diseases?
So that's a very fair question. So there were cohorts in Geneva. So there was a cohort in Geneva. So I'm trying to find it on this. So it's slightly to the right of 0.7 for predicted IFR. And that cohort continued from, I believe, about May 2020 and is just continued through the vaccination period. So that's one of the studies we've included for assessing IFR, IHR, and relative prevalence over time. There were other cohorts in other regions of Switzerland. So probably, to my knowledge, Switzerland is probably, the set of studies in Switzerland are probably the most comprehensive cohorts that were performed early and then continued throughout the pandemic. Other cohorts were started early. Other ongoing cohorts were used, like in the Netherlands. England has multiple. Brazil had not a cohort, but a random sample of the general population that was ongoing. So those cohort studies are very useful. The US was unfortunately was slow. And we didn't have a nationally representative ongoing cohort, unlike in the UK where they had multiple. So,
I mean, I remember early on to try to get death estimates, right? There'd be excess deaths are reported with a huge lag. And so the New York Times was calling funeral homes around the country and the world trying to count how many people were dying week by week to try to assemble those kind of data sets.
Yeah,
I don't know what.
The US, there were definitely complaints that the US was, at least the CDC was very slow in their serology assessments. And when they did perform them, a lot of them were convenience samples and not random samples of the general population. So Brazil was performing those studies in June, Netherlands in April, Switzerland by May, and the US didn't get around to doing it until August or October. So it was very slow.
I guess it would be a little bit later in the epidemic, because they didn't introduce those tests until. How does this kind of study mesh or not mesh with, for example, using wastewater analysis to try to estimate prevalence? How do those? They're very different. They're totally different approaches. How could you put is it possible to put those together to get a better sense of what's going on? And if so, how?
Yeah, that's a great suggestion, and it's been performed in the literature. So there's a study in Jefferson County, Kentucky, where they did just that. They had serological testing of a random sample of the population just multiple times, and combined that with ongoing wastewater surveillance. And they showed that there was a correlation in trends. So I don't want to say prevalence, but as infection trends increased in wastewater, seroprevalence correspondingly increased, and then those increases corresponded with waves in reported cases. So using those two methods together can be very useful. And wastewater surveillance is definitely an important ongoing source of data. It would be nice if other locations continued that as well, but I have not seen as much work as needed correlating serology and wastewater. It's definitely something to explore further.
You mentioned, that this is really a method that's both mainly focusing on primary infection and infection before the development of vaccines. What is it? How maybe I missed understanding how would this kind of test today, when almost everybody in the population has been infected, could it still provide information, or is this fundamentally something that's of value early in an epidemic and that becomes less useful later on? Is there some way, how would I apply it later in an epidemic?
So the issue there is to apply it later in an epidemic, we would need to do another systematic review of seroreversion following infection or reinfection. So it's true that this method does become less useful later in the pandemic, but it's particularly useful for future outbreaks, early in future outbreaks, because say there's a future pandemic with a different virus, the antigen targeting aspect of our work will be very helpful, but the assay design will be. So they can plug in those initial estimates of seroreversion based on lateral flow, indirect or direct, into models to adjust for seroreversion early in the pandemic before they have data. That would have been useful for this pandemic, just to sort of address results like, again, Italy and Indiana. So there wasn't as much of a bias assessment of IFR, because there were a lot of news reports suggesting fatality rates in Italy that turned out to be just not that high.
So that was the next question that I was going to ask, which is, what do we need to do now to be preparing for the next pandemic? What are the specific things that we could do in terms of this kind of study to set those up so that we're ready? Should we be doing, we can't be sampling for viruses we don't know about yet. So we can't be doing those tests, but we could be banking the blood to be ready for it. What would the best sort of public health surveillance strategy be to be prepared so that the next pandemic? You mentioned the fact that you have to have different regional samples. You have to be very careful about bias. What would the ideal, if you had some limited but not insignificant resources, what would the best design be for surveillance and readiness using the information you've gotten from this stuff?
So this is a question where people with more expertise than me could probably correct me and probably give more context. But my initial assessment would be, I have sort of a group of, if I have limited resources, I want a set of manufacturers that have direct assay like setups in place already. So they can just quickly take the virus in question, generate direct assays to be used in the lab, and then start with those because we know those assays are at lower risk of seroreversion, so we're not going to have that bias. Second, we have the ongoing cohorts, which don't necessarily need to be used for just SARS-CoV-2 assessment. They can be used for assessing burden for infectious disease and non-infectious disease. So there are other uses. Again, definitely banking samples so they can be tested early on. I mean, later in the pandemic, because those samples will not only be important for long-term tracking, but they're also important for assessing specificity. So if you have samples from before the outbreak, you can just quickly assess, okay, this study has low specificity, we shouldn't be using it. So that's what I would do if I had limited resources, but that requires buy-in from companies, because one reason why so many companies use lateral flow assays as opposed to direct ELISAs is because lateral flow assays are cheap. You can just market them to people who are outside of a lab. They're happy to be able to use a test even though the results were less reliable. And there's profit there. So there would have to be some sort of institutional push to encourage the use of direct ELISAs and have those frameworks in place quickly for a pandemic. I don't know the politics of that. So
I guess I was thinking more on the cohort assembly side. How do you build and define the cohorts you're going to be sending out test kits to people and having them report is one thing. As you point out that there's all sorts of biases if you don't have a pre-selected cohort. And you mentioned the possibility of there being, if it's geographically isolated, you might miss things.
Right.
What if you, in the US, for example, I mean, would you have a sort of a sentinel cohort in New York, a sentinel cohort, and how would you do it? Where would you pick the people, I guess is my question.
That's a fair question. So the UK has a fairly good model of this even before the pandemic. So they have the UK Biobank, where they had just an ongoing cohort of adults across the country. So the US could presumably have a similar cohort. I don't know why we don't. I'm not in a position to say why we don't. But that would be useful, because if you say this is a problem we ran into early in the pandemic, where there were specific studies, say in California or in specific regions of Germany that were brought up, that were small populations, they just did not give accurate assessments for later in the pandemic. So a national cohort is necessary, not just a small local regional one. You can have regional cohorts like we're in Switzerland, but they had regional cohorts from multiple places in the country. So it just wasn't only Geneva, they had one in other cantons of Switzerland. So if you're going to have a regional cohort, then it needs to be multiple regional cohorts in diverse populations, rural, urban, different racial makeups, different age makeups. Because if it's just like in New York or just in one small city, it's not going to be helpful.
But presumably both hospitalization and fatalities are determined not only by the disease severity, but by the availability of hospitals, the quality of care in the hospitals. There's a whole variety of other factors. So you'd like to sample across that as well as the patients. Okay, other questions from the audience? Lucas, are you on? You have connection? I know you said in your chat notice that you were disconnected. Did you have anything you want to add, Lorenzo?
Well, This is very much outside my field. I don't have any comments on this talk. Sorry. It was very interesting though.
So one area of concern for me is sort of serology results versus modeling results. So there's been a lot of suggestion that early pandemic modeling from, say, the IHME or Imperial College was inaccurate and sort of gave people doing pandemic planning inaccurate information. And so we can use serology to assess that now. So we can take serology results and compare it to the estimates that we're giving by using models early in the pandemic. And the argument I would make is that the modeling was actually quite informative in the absence of serology, and that serology sort of provides a balance with modeling. So modeling can give you results very, very quickly while it takes a while to run serology studies, but also serology is susceptible to biases that modeling is not, and vice versa. So there were some early serology studies with non-random sampling, using assays with bad specificity or at risk of seroreversion that were clearly worse than the modeled results from very limited data. And so one suggestion I would make is that in future outbreaks, we take the modeling results quite seriously until we have serology data. Because again, my overall impression, I'm working on a piece on this, my overall impression is that the serology results from random sampling can largely confirm the predictions that were made in March 2020 using models. And so it would have been nice to have more attention and pay to those things.
So maybe as a final question, then, if just on the time scales. So to have the serology test work, you have to be able to have some test that works to detect your virus. You need your ELISA in place. About what if virus X shows up, can you walk me through quickly what the sort of the delays are? Virus X shows up, it's identified as a virus of concern. Maybe initially it's not even identified. You have to identify it first. And now, what's the time it would take under best case situation before we can have the serology information that we could use to inform public health decisions?
So if we use COVID-19, this pandemic as an example, we had reliable, probably the first reliable serology result I can think of is late March, early April, with a reliable assay on a sample that's random sample representative. So, and I think COVID really started to be taken seriously by mid-January. So that would give you two and a half month period. That could definitely be sped up if you have sort of a testing framework already in place for developing the needed ELISAs. But I think it would take 2 months, because you have to again reach out to the people to get them to use the test. If you're doing an ELISA and it's in a lab, it's going to take more time. If you're using a test that you're sending out to people, it'll take them time to send the results back to you if they're using like a lateral flow test or something. So I would say, based on the COVID-19 pandemic, 2 months is probably the earliest I could foresee. 2 months from when people really start to notice it and take it seriously.
So Lucas had a question in the chat, which was, are you aware of any ongoing projects that try to assess the fatality associated with antimicrobial resistance? There was a study in Lancet last year, but there seems to be relatively little work done in this field.
Antimicrobial resistance, not that I'm aware of. That would be definitely something interesting to look into, but not that I'm aware of. You could arguably use a cohort for that, but antimicrobial resistance isn't typically more in antibody use in like hospitalization settings? So you need a cohort with a fairly large hospitalization component. So I'm not aware of any cohorts like that.
mean, I guess one thing that might come up, and again, I guess it depends, is, well, no, I was going to say, I mean, when I see something antimicrobial resistance, I think things like tuberculosis. And there, I don't know what the serum signals are compared to the actual latent infection. You can have latent infection that lasts for a lifetime. I don't know whether you're antibody positive for a lifetime. I guess some antibodies are persistent. I don't know.
And it would likely be also vary by assay as it did for SARS-CoV-2. So we probably want to select for an assay where the limit of detection is high enough, not high enough, low enough to pick up those antibodies.
So would this kind of testing and response be able to give us some estimate of vaccine effectiveness, or is that something that's outside of the remit of what you're doing here? We don't know if the antibodies are induced by the infection versus the vaccination, or you did distinguish the antibody signatures.
So the antibodies that are generated from infection can differ from the antibodies that are generated from vaccination. So infection is going to generate antibodies targeting nucleic acid protein, while the standard vaccinations won't. They'll only generate anti-spike or anti-RBD. So you could use that to distinguish infection versus vaccination. The issue is vaccination is decreasing the sensitivity of the anti-nucleic acid assays, largely because my hypothesis for that is that it's promoting, there's sort of competition between immune cells, and the vaccination is promoting immune cells that generate antibodies against spike or RBD, but not against nucleic acid. That becomes really important because people have been using anti-nucleic acid assays to track infection post-vaccination without accounting for that adjustment. So I think sort of a really good example of this is there have been suggestions that children have a higher infection rate. I said before that infection rates are lower, were thought to be lower in children, but there have been other suggestions that infection rates are higher in children, or prevalence is higher in children. And the problem with that is they were relying on anti-nucleic acid assays, and children weren't vaccinated yet. So children having higher anti-nucleic acid prevalence was just due to them not having the vaccination that reduces sensitivity. So once you adjust for that, there was virtually no difference. So I'm really hesitant about using this framework to assess post-vaccination unless one modeling is good enough to account for that, to account for vaccination effect on sensitivity. Otherwise, the conclusions reached will just be not informative.
I guess I was wondering, not so much for SARS-CoV-2, but in thinking back to something like tuberculosis, of course, very different. But one of the problems, I mean, there's been a tuberculosis vaccine was one of the earliest vaccines out there, but it's extremely ineffective. Existing ones are extremely ineffective. And I wondered whether there's anything you could do to try to basically, I guess you need very large populations if you're going to try to get some estimate of how do you assess vaccine efficacy? Can this help you assess vaccine efficacy?
So, I mean, in the context of a virus, it'd be more straightforward because then you could just run a neutralization assay. And then choose a neutralization assay that seems to be tracking antibodies that aren't seroreversion isn't as bad. But outside of neutralization assays, could you use this framework to test? Yes, but the most it would likely tell you is not to use lateral flow assays and probably focus on directs. And so, and the problem you're going to run into there is that your direct assays are going to pick up non-neutralizing antibodies, which aren't the antibodies you want the neutralizing ones, at least in the context of a viral infection. And I know TB is like TB is like largely avoiding antibodies by hiding within cells, so it makes it more complicated there as well.
Again, I mean, I didn't want to focus only on TB, but it's a case where I know there's a lot of new work trying to develop more effective vaccines.
Right.
So historically, of course, the vaccines for TB were not used in the United States. They were used in France. And so you had populations that had very different vaccination profiles. I always am amused because I've been vaccinated for plague. People in the United States don't even know there is a plague vaccine. That's a Russian vaccine. I have no idea how effective it was. But
I know that.
there are these regional differences in vaccination profiles and adoption, which could be again, if it's a virus X that's not known, then of course there's no vaccine, so it's not an issue for you. But
I know there were, I don't know, I haven't followed up on this, but there was some suggestions that BCG vaccination early in the pandemic might have had effect on fatality risk from COVID. And I can imagine a scenario in which vaccination with against a related pathogen could influence seroreversion or antibody titers against a newer pathogen. So if there's, like, for example, another coronavirus pandemic, would the vaccinations against SARS-CoV-2 impact seroreversion against those newer coronaviruses? So it's an interesting question, like the dynamics between vaccination and then subsequent responses to infection. It's just difficult. It's difficult to assess. And I'm not sure how this framework would help in assessing it. But it's an interesting question.